Hi everyone. Today, welcome back to the channel. Today's topic is Lab Exam 2 Study Guide. Okay, so I'm gonna go over the major labs that we cover, and then that will be on the Exam 2 this this coming week. Okay, so first there are 6 lab topics that will be covered here: secondary growth, plant growing, leaf, flowering, plant ID, and surviving. Okay, so we start with the secondary growth. So what is secondary growth? This is there are a number of terms that they need to be defined, including vascular cambium, core cambium, tree rings, annual rings, 3 types of cuts: spring wood, summer wood in a single year cut, and a plan of the week of that specific week. Okay. The next lab is plant growing. This is just basically, there was a video involved with this, and it's always remember cell enlargement is the major contributor to the plant growing, more than even plant cell division. So terms like height, tree age, and the questions like what are the adaptation of certain plants to the certain type of situations, and then they need to be actually studied in this. And then plan of the week here, which is Colliers. The next lab is leaf lab, which is an important lab because it actually covers leaf anatomy, leaf morphology, leaf distribution, and a little bit about photosynthesis, because actually anatomy of the leaf, which basically also determines photosynthesis type, or vice versa. We have 3 types of leaf anatomy. They are C3, C4, and CAM. Basically, these are based on the photosynthesis types of C3, C4, and CAM. If the leaf morphology, there are single leaves versus compound leaves, and leaf can be sessile like no petials, or bladeless like no petial, and no blades, I'm sorry. And then they can be compound leaf, which have leaflets. Okay, a typical leaf anatomy you see here, a typical C3 leaf anatomy, including a palisade mesophyll layer, which doesn't exist in C4. And of course, the stomata, the spongy mesophyll, lower and upper epidermis, cuticle, and guard cells. And then plan of the week for leaf lab. Flowering lab, which starts with basically microgametophyte and microgametophyte and megagametophyte, pollen versus embryo sac. And then parts of flower, which 4 major parts: SP, SC, sepal, petal, stamen, carpel. Right, carpel is also same thing with pistil, which is the female part of the flower, which has stigma at the top, style, and then ovary at the bottom, which becomes fruit later, and ovule inside, which becomes the seeds later. Right, and then we have a number of questions about the largest flower and others, some assumptions about flower color. Always remember red flower more attractive to the birds, but not the yellow or white flowers. Next lab is surviving lab. This is about plant survival. Basically, so first we need to know what are the 4 things plant need for survival, and also some environmental conditions actually we discussed that actually limits the survival, and how plants actually very flexible, and they are very resilient, and they survive in environments like that. The next lab is about plant ID. We have 2, 4, 6, 8, 10 plants here that you need to know their scientific name, their common name, and their class, right? And then there is a plan of the week there. And always remember our monocut model plant, which is maize, Zea maize, and dicut model plant, which is Arabidopsis thaliana. And also we divide plants in 3 groups in terms of terms, annual, biennial, perennial. Perennial plants have some advantages that they don't need to be replanted every year, like annual plants. And biennials are very interesting because they actually, their life cycle takes 2 years. This is it for the exam, lab exam 2 review. Thank you for watching and happy studying. See you next time.